Okay, transmembrane proteins. Let's try that again. So the problem is when we use the regular method that predicts transmembrane proteins, we get a completely wrong prediction. When we use some hydrophobicity index, we can get some prediction, but we'd like to do better. We like to do that better by simply training the same system or the same type of system that has been described before to predict secondary structure, now to predict transmembrane and non-transmembrane helix residues. So instead of where in secondary structure we have a 3-state prediction, now we have a 2-state prediction, transmembrane not. Again, there's a 2-layered approach here. The first layer has helices, transmembrane helices, range between 17 and 25 residues typically. On the first level, you predict things that are much shorter than that are sub 10 residues. And that's why there's a second level introduced, and that makes helices much too long. Now the average predicted helix is getting to the values of about 40. So this is too long. That's why we want to cut. And the idea of the cutting algorithm is we take out directly the output, we read out the output from the neural network unit T and the neural network unit N. Again, you could code for this problem using only one unit. 2 units buy you a lot of advantages. Essentially, the advantage, for instance, is that it's better to distinguish, or it's more potent in distinguishing the reliability index, for instance. Okay, so we have the signal here plotted along the sequence, along the protein sequence where the first residue is here and the last residue is here. And this output lane is here. Now, since these two, you sum them up, and then they sum up to 1, you normalize them, and that means that essentially the information is redundant, that you can get away with one of these pieces to describe the prediction of the neural network. So that's now the description of the landscape of what the neural network predicts to be membrane helix. That's the image I showed you. Here's sort of an hypothetical image that I made up. It could look like this. Now, if it looked like this, so the typical threshold for the decision making here is 0.5. Everything above 0.5 would be predicted as a transmembrane helix. And you notice the numbers here are running from 1 to 100. So you do see that this region that is above 0.5 here would end up to be something such as 5 to almost 60. This is much longer than a transmembrane helix between 17 and 25. So that is exactly the kind of example where you would see that the transmembrane helix is predicted much too long. Now, how could you cut? So the simplest algorithm would be to simply cut in the middle. Because you know it's twice as long as you want it, essentially you cut in the middle. And that's a hack, in fact, that I published at some point, but you know, you don't want to live with hacks like that, so you try to do something that's a little bit better. And a little bit better sort of is this greedy algorithm that I try to explain. The greedy algorithm begins by enumerating all the segments. And what I'm showing here is for this curve here, this prediction, this hypothetical prediction, I show all the segments that are either 19, 21, or 23 residues long, for which the average score is above 0.9. And I just show them all here. And I sort of loosely put them somewhere next to the hill, because I cannot put them in the right place. There's just not enough space. And you notice that one of them here is in fact not 0.9, it's a little bit lower. But for this function here, or for this prediction here, those are all the segments that are either 19, 21, or 23. So again, typically I sample 17 to 25. This is just for simplification here. The complete set of all of these segments that are high, that have an average higher than 0.9 are shown here. Okay, and the way the algorithm works in the greedy approach, it simply picks the highest value. Now, in this particular choice, you see that there's a bunch of highest values. This 0.94, 0.94, 0.94, and 0.94. Typically, the way you will program it, most likely your program will take the first. And this is ultimately what I do here. If you don't consider that this could be an option, if you consider it to be an option, maybe you make a cleverer choice. But one choice would be you take simply the first number, okay? The first segment that is the max, you take it. And that would be this. Then you take the second best, In fact, in this particular case, the second best is as good as the first best. That would be that. And then you take the third best. And actually at the choice of the third best, you already see that there is no third best. And again, so the way I list these numbers here, that means the segment would run from residue 28 to 51. And you see this runs from 37 to 56. So they would actually overlap. So they cannot be both chosen. That means that, in fact, when you look at these numbers here, none of these numbers, so again, what I'm showing here is the entire segment of thing, or the entire set of all possible segments above 0.9, and there's no other choice here that is above 0.9 already. And in fact, I had sort of to dig deeper into my file, and then I get to 0.87. That's the next highest value. Is the fragment or the segment between 15 and 34. And in this particular case, you cannot find, when you look at this function here, you cannot put anywhere else a transmembrane segment that is at least 17 residues long, that has an average over 0.5. Yes, there are still regions left. If you took this one and this one, there's still bits left here that are above 0.5, but none of them would add up to another membrane helix. Is the algorithm clear? To everybody? No.
Isn't this like a one-dimensional net stack problem?
Yes.
So is the greedy approach really the best one?
No, it's not the best one. This is the greedy approach is the best one given the limited time that you have for this choice. So ultimately, the question now begins: how much do I do? So the simplest thing is you cut it through into two if it's twice as long as I expected, or something like that, right? Or you cut away if it is too long, and not twice as long, but little. You just cut away. You just take the peak of the 17 to 25. That's one simple way of doing it. Now, while that is simple, it's sort of, if you publish this. I published something like that because I did that. I had to send it tomorrow morning, and I just had a few hours to do it. So then you can do that. But if you publish that method and then you sort of put it online, this is not feeling comfortable. This is not something that you are happy and proud that, I did that. So at least you do sort of the second best. The second best is this one. Now, optimizing the optimal path through all possible ways here is CPU intensive. So now we get into the point, how much do I gain from this cutting business? So if this cutting business takes as long as with the prediction business, then it becomes an issue of what is the value of what you add. So the greedy approach is good enough because it adds a lot of value for a very short CPU moment, right? The real algorithm, the optimal solution, is not gaining enough for the CPU it adds. That's as simple as that. So sometimes simple solutions, in fact, it is much better than the simple solution where you have empirical values to cut. Now there is a step that is still left to be done. And that step, remember I told you, we also want to predict decidedness, the orientation. The orientation is which residue is where the protein starts. What's the first residue of the protein? Is that sort of outside of the cell? Or is that inside of the cell? Is that the beginning or is that the beginning? So from this image you cannot see that. It would look exactly the same way. So this is something that you really have to predict which way it is. And I said there is from Gunnar von Heinje this positive charges inside rule. So what we do is we take all odd loops. So as loops I consider something that is next to the transmembrane helix. And I look up how many positively charged residues do I have, and it's the sum for odd. I take the next odd one here, number 3, I do the sum, and then I do the sums over the even ones, over the even residues. Okay, in this particular case here, I would have 5 plus 1 for the even, so 6, and 2 plus 3 for the odd ones, so that 5 is smaller than 6. I say positively, there are more positively charges at the inside. The balance in this particular case is a close call, but nevertheless, we assume that therefore these guys are sitting in the inside because the number 6 is higher. Okay? The even loops are these here, this and that. Is that decision clear? There was a way to mess up this image again, right? Okay, this one, this one did it again. Hi. One image typically gone over in 1 minute, and okay. So we have 1, 2, 3, 4 loop regions. So in this particular case, what I mean by loop regions is these are regions that are just next to the transmembrane helices. Right? They're not buried in the whole protein, and they're not in the membrane. They're just at the edge of the membrane. And of these we have sort of 1, 2, 3, 4 regions. Okay? What I know from the observation, in these regions here, if I look at all the positively charged residues, and there are in fact 2 amino acids, R and K, that have positive charges. So simply ask yourself, how many R's or K's do you find in this one? How many R's and K's do you find in this one? You sum that up. And then you ask how many R's and K's you find in this one and how many R's and K's you find in that one. And then you know that the one with the higher in the difference. So I'm just looking at the sequence, and I ask whenever in this sequence is the residue R or K. And that gives you a delta function of 1. Okay? And I sum the R's or K's here, and sum the R's or K's here. And that's one side of the molecule. And sum the R or K's here, and sum of the R or K's here is the other side of the molecule. Now the question is, which side is inside, here, and which side is outside? And again, inside is the one with more R and K's. In this example that I show, more R and K's, namely 6, in this example shown, are here, and 5 are here. Or is it that you don't get it because I made a mistake? 2 plus 3 is 5. 5 plus 1 is 6. So it's not my mistake. There's something that is not getting through. Can somebody help me out what it is that is not getting to you? So R and K are simply 2 amino acids, yes?
I don't understand what you mean by even and out.
Even loops. So this is loop number 1, this is loop number 2, this is loop number 3, and this is loop number 4. 1 and 3 are...
Okay.
What? Is that what everybody is stumbling over? I see that I have essentially an entire room full of question mark. So if you don't get it, the one next and left and right of you also doesn't get it. You all look the same way at me. I do not know what is wrong. Yeah?
So R and K are parts of these loops.
Yes, we have 20 amino acids. 2 of those are, one of those is an R and another one is a K. So we have 18 other amino acids. And I simply count because they are the only ones that carry a positive charge.
Is there a difference if I count an end or a loop?
And what
Get up there.
sorry, N, what do you mean N?
An end.
End. So yeah, so that's a very good point. So if the end would be such that you actually end with a membrane helix, that would sort of kill it. Typically the end is a little bit more. So I do not believe, yes, I've seen cases where it really ends with a transmembrane helix, but it's a very rare case. And if the end is a transmembrane helix, then my story about odds and even will change. And you clearly will not get a sum of 1 here. Because if it is 0, you cannot get 1 R or K in a set of 0.
Can you show us maybe where you get these numbers from?
So yes, I'm sorry, these numbers are taken from above. So they're completely made up in this case. So I should have given you a sequence. That's right. Now I'm going to take also, that was the last. Let me make you understand one thing. I'm absolutely grateful. This is great. It's the first time I've given this to more than 5 classes. You're the first time that I'm stumbling over this image, and I'm really grateful to you. Yes, I feel strange, and yes, it's more work. But as long as it works, to make the story clear, I have gained. So I thank you. I really mean it. It's not lip service. I'm really, it's also, I don't know how often it happens to you, that you have done something for 5 years, five times, 6 times, and then suddenly you realize it was actually wrong all the time. Just like sometimes there are people who spot typos. After years of showing slides, people spot typos. Incredible that these things happen. It just tells you how difficult it is to get the detail right. Anyway, I will show you the sequence. You cannot see the number of R and Ks. At this particular case, are just made up numbers to illustrate that. And in fact, it's true, I should have made up numbers such that the difference is a little bit more extreme. So the 5, 6 is a very close call. So they were in fact not made up. As all of these examples before, they were taken from a real example. It becomes much simpler to explain things when you completely make up a case. So I completely invented the numbers. That is this example here. Okay, I'll show you the sequences next time. So essentially, if I had sequences, you would see that in these 30 residues, there are 5 of these 30 residues are OK. Which in fact, as you compute probability, is unlikely, right? It's much more than random. But anyway, this is that's where the numbers come from. So again, you have a sequence running up there, and you simply ask, there are 5 residues in this part here, there are 30 here, there are 6 here, and there are 5 here. And now you ask, of these 5 residues, how many are either R or K? And the answer to that question is 2. How many of those 30 are R or Ks? 5. How many of those 6? 3. How many of those 5? 1. If you have these numbers, you simply have to add up what is on that side of the molecule and add up what is on that side of the molecule, what is on that side of the molecule in terms of number of positively charged amino acids. Yeah?
So is the inside of a cell more negatively charged, and that's why it's like the positive part is inside?
Yes, that's exactly, but actually that is true. And this gradient effect is important for the transport of molecules into cells. And it's slightly also different within different compartments. But essentially that is the truth. What is weird about this truth is you could imagine that you essentially also created by having fewer negative charges, right? And funny enough, the number of negatively charged residues is exactly balanced between the outside and inside. And given the excess of negative charge inside, you would not expect that. Right? You get my argument.
Like the difference between the number of R and K is like equivalent to the effective charge?
No, I believe I was encouraged by your question and I got too complicated in my answer. Encourage is the wrong word. Something in your question made me become too passionate, or whatever you call it. So, yes, indeed, there is a tendency for more positive inside the cell than outside. So there's an excess of positive, the other way around. Positively charged residues because the environment is more negatively charged, which is exactly your point. So there is more negative charges here. And again, where's the 5 coming from? I'm just making it up. I'm just trying to say there are 5 negative charges here, 3 positive charges. So the inside, the solvent inside of a cell is more negatively charged. This is why you would expect that you have in a protein more accessible negatively charged amino acids, positively charged amino acids. Right? This is exactly what you argue. And you're absolutely right. Now, if you argue that way, then you will say, okay, so the positively charged residues are R or K. The negatively charged residues are Q or E. Given that reality, it works out so nicely for the positively charged residues, it should actually be visible in terms of the balance of the negatively charged residues, right? There should be few of those, and that is not the case. No. That's totally unexpected. So the first part of your argument, of your question works so brilliantly that you're very surprised that the second part doesn't work. And since the second part doesn't work, you begin to wonder whether in fact the first explanation was right. But it's true. These numbers appear to be matching. But the negatives are...
The protein can also be negatively charged because it has a lot of QMEs in it.
It's true what you say, but then you could make this... The moment your explanation no longer holds for something else, that is a good way to get out. But if that is a good way to get out, you have to sort of doubt your previous explanation, although it did fit.
Yeah.
That's what I'm trying to say. I'm sorry.
So what's the explanation now?
We actually don't know. It remains a puzzle. We actually really don't know. So when that was found that for a large data set, so at the time of Gunnar von Heinje, he just had a statistical signal for the positively charged residue, and he never understood it. And 15 years later, in fact, I sort of rechecked with a much larger data set. I clearly established that there's no signal for anything else. And that is a puzzle that remains.
Always it more RNKs are in the inside, or is it like mostly?
Say again?
Is it always that there's more RNK residues inside, or is it like mostly more inside?
It's not. This is biology. This is not always. And immediately you see reasons why. Because if you have DNA somewhere, so they are also DNA binding residues. So you have many functional reasons to knock out rules, so to speak. But the overall average signal is such that you can use it to predict. This is good enough as a statistical signal to allow you to predict.
Is it like for 90% of the cases? Or
it is, in fact, so I cannot remember what, I believe it would be in the ballpark of 90%. What I cannot remember, so I do not know the answer in terms of numbers. What I do remember is how much, if I use that signal or I don't use that signal, how is the topology prediction going to change? And that is in that ballpark of 90%.
okay.
So I have to think it through a little bit more to see whether I can I cannot directly translate these numbers. I could check, I don't know. But I still have the feeling, does everybody get this thing with the positive and negative charges now? And I just will not look at you unless you say no. And we move on. Because I want to get through with the membrane proteins. And I'm going to rush through something else. What you get is a prediction that does this one. So where you previously saw the blue, you now see it's all red, so the prediction is completely right. You could also apply a different type of machine learning device. In particular, in this case, hidden Markov models are the solution, and this one comes from Anna Schroch. The beauty of the hidden Markov model is that the whole problem has a grammar to it. You have a part that is the helix core. You have a cap, so at the end of the transmembrane region, there's some reality. Then there is a loop, and then there's an entirely globular region. And a similar story on the other side of the molecule. So you have different situations inside the lipid, at the edge of the lipid, and outside. Yes?
Can you briefly discuss the page before? Like, there's just one ST, right?
There's just one T.
Yeah, that's like, I don't understand what this means.
The T, that's absolutely consistent. That's the T that you asked the last time. It's the same T as before. It's transmembrane helix.
Okay.
I'm sorry. I apologize the last time to you asked the question. I'm switching the notation here from T to H, or H to T.
So this from OBS to PhD just gets one less transmembrane.
So it's essentially, I replaced the H for transmembrane by a T. And the reason is that once I had this method, I suddenly needed a method that where I can look at transmembrane helix and globular predicted helix, because I have two methods and I actually map them to the same sequence. So I introduced a new symbol, historically.
And what does this slide say?
This slide essentially says that we have a protein here, the protein sequence, and that protein sequence is observed in a transmembrane helix here. And that is the prediction of the transmembrane helix. So essentially in terms of accuracy, there's one mistake. And this is the one where I previously showed you that this was entirely blue. Bless you. Was entirely blue. So the prediction previously was very poor, now it's very good. to say the least. Here's another way in which you can do that, I said, with a hidden Markov model. And the beauty of the hidden Markov model is that you have a different biophysical reality for different regions. And this different biophysical reality in the hidden Markov model is simply modeled by different probabilities. So you have essentially different modules. Whether you enter in your chain, whether you enter another cap, or you get into a helix, or whether you go to the loop, all of these transition states are things that model the type of event that happens exactly at that interface. So hidden Markov models are optimal for this type of problem, and therefore they have been used for transmembrane prediction and are very successful. And I'm going to show you another example in a few minutes. There is one issue about how we in fact estimate or how we measure that the performance is correct. So here I have an example where we have 1, 2, 3 observed helices, and we have 3 predicted helices. And now one way of counting would be to say, well, I have some substantial overlap with this helix, so I call that one right. I have some substantial overlap with this one, I call that right. And so forth and so on. So you could essentially say you get the number of helices right, and you get essentially the placement right, so it's an entirely correct prediction. Or you could argue, we discussed that already, that you allow only counting once. So if you count this predicted helix as correct, you count it either here or here, but you cannot count it at both places. And in some sense, the same is here. So if you count that, if you consider that the overlap between P2 and O3 is high enough to consider the correctly predicted segment, then you can count P2 but not P3. Because that would P3 overlaps with the same segment on that side now that you already counted. And if you allow less overlap, then this gets more stringent. This is a relatively stringent way and looks at a bunch of membrane proteins. The measure here for success is essentially if the overlap is at least 3, they can differ maximally by 3 residues on either side. If you predict something that is within less than plus minus 3 residues, then you say it's a correct prediction. And the number that I'm showing you on the next scale, bless you, is 1. If for the entire protein you get all the helices right. So you see that for different methods, the number is in the ballpark of 0.8, the estimate. So later we sort of found out that this is a slight overestimate, and that for newer proteins, we see problems that we haven't anticipated before. So one problem that we, for instance, see is helices that in fact are not really transmembrane, but they sort of hover at the edge of the membrane. So they go into the membrane region a little bit, they are hydrophobic, but they essentially sit more parallel to the protein than going inside the membrane. And of these, previously there were very few known. Now it's we over the last 15 years, more and more of these have been discovered. That's one problem. Another problem is this one here. So you see this blue helix. The only reason why it is blue, it is a transmembrane helix. The only reason why it's shown in blue is just to highlight the problem. Can you all see the problem? Yeah? It's too short to be fully transmembrane, but if you you see that twice, right? There are 2 that are too short to be fully transmembrane. And it looks sort of, if you look from further apart and you don't have good glasses or eyes, then it looks almost as if this is one transmembrane helix. So with a little bit of fantasy, you could see this as a transmembrane helix. And in fact, in terms of structural stability, it acts like one transmembrane helix. But it's 2. And this, of course, for all these kind of algorithms that I talked about, a transmembrane helix is 17 to 25. Screw you, right? Sorry. This is not going to work, right? The method that we had such a trouble to understand is going to completely fail on this, yes?
Normally they're capable of transferring things in and out. Can this transfer something? Maybe it just transfer something in the fatty region? No,
so in this particular case, I actually don't know what this molecule is, but in principle it could still have a hole inside. You would have to turn. So the way they are tilted, it could be. You would have to sort of turn it and see whether there's a hole. But it is also true that very often the function of transmembrane protein comes by aggregation of several units. In which case this would most likely then only be one unit. Not all, also the issue is, I'm not sure who said that in this previous issue, but many of the transmembrane proteins in fact are anchors. They're sitting there and they recognize some event, and that some event is triggering, and that some event triggering means you do something to that loop that will induce a slight mechanical strain. That slight mechanical strain will be visible here. So essentially we'll push a signal through without pushing any material through. There's another example how structure is important. So it's the rigidity of the structure that allows you to sort of change something here, and since it's so rigid, this will have an effect on that side. Okay, and this could be that too. I really don't know what this one is doing. Okay, but this is another issue, and ultimately there's a bunch of other issues that they come at different angles. So this generic way of looking at it where they are orthogonal to the membrane, there are more and more examples where they're really tilted very strongly. They end at locations that are not, so this is sort of the one that is hanging on the membrane somehow, but there are more and more cases where you begin to see that here, or begin to appreciate to see it here, where this one is not really getting to the end. I said helices go through, this one is not really getting through, yes?
So you need this whole thing to let a very specific molecule pass through the membrane, right? Because the helices look like pipes.
Yes, but again, no, don't think about the helices as pipes. The helices, whenever you transport material, it's not going through a helix. So the way to think about it as a pipe is that you have 5 helices, and the 5 helices form a pipe like this. That's what you typically, and in most cases, in fact, it's the 5 helices, another 5 helices, and another one, and they form a ring. And that is typically a big gate. Many of these segments coming together.
Is that the scale for the material where they have different forms?
Yes, exactly.
Okay.
And sometimes the shaker channel, potassium channel, for instance, is a case where the angle of the transmembrane helix, and this is a little bit like the example that I showed you before, although it's a much bigger structure, but it's a little bit tilted like this one, where in fact you can filter the potassium exactly by this angle. So this angle gives you a very fine controlled ability to open and close and open and close and be exactly for the right potassium. So you have the right chemical charges and the amino acids there. But it's something that looks a little bit upon first sight like this one. Again, I agree with you. This may not look like it transports anything. But it's never inside, it's never through the helix. Okay, so there are many more problems from the later structures, so from the most recent structures that have been done, and these lead to new methods being developed. One of these new methods here tries to in fact not only say there's a helix, where the helix is, how it's oriented, but also how much this helix, where in this helix are you? Are you in the middle of the membrane? Are you sort of toward the end? Hoping that we can use that again, first of all, once to predict 3D structure, but also possibly to see whether that helix is really entirely in the membrane or not. So this, again, is machine learning based. That is one way. So the original idea was that I would be talking about another method, TM-sec, that employs in fact random forests to do this, what we said before, better and faster, but I'm not going to do that. I'm going to skip all of these slides. So TM-sec is a random forest that does what I said, what I showed you before, and it does so much faster and with fewer, with a better performance. But this will not be part of the exam. I will just skip over it. There are proteins. I will just show one example that use beta strands to go through the membrane. In fact, these are much bigger things. So the strands, so each one of these here is a strand. The membrane is indicated by these dashed lines. So that's the top, that's the lower part of the membrane. You see this part here is sitting in the membrane, right? And the way they sit in the membrane is every finger, imagine every finger here is a strand. And really these strands are at a 45-degree angle to the membrane axis, and they form a hole. And they really punch holes into the membrane. In fact, most of these are passive transporters. Passive transporters means essentially everything that has the right, or that is not too big, goes through. Proteins don't go through, but smaller things, when sort of if you unfold a protein and have it as a long line, you can put it through. So they are relatively large barrels. And again, you can predict that in this particular case, Markov models or Bayesian models are best. You can also predict where you are in a protein, on the outside or the inside. That is a prediction of solvent accessibility. And this would essentially conclude the idea of 1D prediction, except for the issue of disorder. So the credo has been in this talk that structure determines function. That's why getting the same structure is always important. And today, I'm going to kill this statement. So typically, order is the thing that is important for structural biology. But then there are things that are sort of weird. And these things are things here. Trypsin is a molecule inhibitor. The inhibitor of trypsin is one that is sort of unstructured. Unstructured means that, so a typical protein having always the same structure means you put light at this object at two different time points, and you observe exactly the same thing. Now we have cases here of proteins, you put light at two different time points, and you do see different things. You see a lot, you could call that a lot of dynamics. So I also told you that proteins are very dynamic, and function is in the way they are dynamic. But this is more than that. I do not even see the same thing twice. So I see a constant change. And these kind of, there are different words for what that is called. Protein disorder is one. Natively unstructured, natively disordered. There are many, many intrinsically disordered. There are many words out there because this is a phenomenon that has been observed. First, in fact, through prediction methods, that there are regions of proteins that are strange. Then it was sort of defined better. Strange means it does not adopt a regular 3D structure. And then methods began to be developed that predicted. One way of predicting is B-values. B-values, and that goes back, or Predrag or Radivojac was one of the people who made the first step here. When you look at three-dimensional structures, this is an example here, lysosome. You sometimes, when you look at NMR structures, you see many structures. I've shown you that before. Many structures, so every line here is one structure, and there are regions where they all occupy the same point in space, and then there are regions like this, this, or this, where they do occupy different areas. And that essentially is typically interpreted to mean where you have different solutions, where you observe different things for the same protein, are regions where the protein moves a lot. Where you always see the same thing, the protein is very stable. Okay? And ultimately, in the extreme sense, now imagine a protein where the whole thing is unstable. That ultimately, where every single time you observe something different, that would be protein disorder. But before we get that, what we could predict in principle is which residues are the ones where you will see more motion, and which are the residues where you see more stability. And that is called B-values. Can you sort of predict from sequence alone how flexible a residue is? And again, machine learning does this. In this particular case, the trick is the normalization, or the way you learn that. Maybe this is also something that one should redo. Actually, we are redoing it at the moment. So this is a normalized B-value. This is very flexible here, and this is very rigid on this end. And this is a distribution over 1,500 proteins. So these are proteins for which I know the three-dimensional structure. From knowing the three-dimensional structure, I know which residues are more flexible, which positions, okay? And what you see is, it's difficult if you wanted to predict this function, yeah?
In multiple proteins for each amino acid. So
Yes.
amino acid will have the same B-value in all proteins.
No. So the question is, will the same amino acid always have the same B value. Absolutely not. Even you see here that some fragments of helices are very rigid, and some fragments of this helix here, going here, this gray part here, that's a helix. If you don't see it, just believe me, this is a helix, and this helix here is much more flexible than this helix. And it could very well be true that this one here is a leucine. I make that up, of course. This is a leucine and this is a leucine. So there's two leucines, both in helices, and one is flexible and one is not. And they are even near in space. And this is a very, very typical situation. So no, the flexibility, what do you believe will determine the flexibility? If it's not, so yes, when I want to predict it from sequence, there must be some sequence signal. In that sense, you're right. So some amino acids are more likely to be flexible than others. That's true. But ultimately, whether that particular L or that particular amino acid is now flexible in that position and not flexible in the other one, what will decide on that? Yeah?
Maybe the way it folds. So,
the way it folds.
So like there are interactions with the rest of the protein.
So there are interactions with the rest of the protein, and that's the crucial bit. So it's 2D, it's contact or a distance map. So the more, if you look at this particular residue in the space of its structure, so the question is, how many interactions does this particular one have with its environment? How much is it constrained? The more interactions it has with the environment, the more it's constrained, the less it can move. Essentially, this is like you're crammed into an elevator or a subway, as opposed to having a lot of space, right? So if you make a photo in a camera in the subway from a seating of a person over 1 minute, and you make the photo of the same person being here giving a lecture, then the degree of change of movement in a completely dense subway will be completely different from the degree of movement here, right? Simply because I can. Well, okay, there are people who give the entire lecture like this? Are there people who don't walk? I walk a lot. Are there people who do not walk at all? But then they are there, right? Yeah. Are there people who stand here and don't walk? Okay. Well, it's an interesting place. Anyway, you will hardly see that with me, except for I break my fingers. Anyway, yeah?
When do you see this flexibility? Is it when it's folding, that it always folds?
No.
Or
So
like after it's folded, it still moves?
Exactly. So the question is, when do you see the flexibility? When will you actually see how flexible a residue is? The question is, during its folding? No. You will only see it when it's folded. You're right. At the end. Because at the end, that's the moment when all your constraints are there. At the end, when you have the final state, the final 3D structure, at that point, you know all your neighbors. That's the moment when you know all the distances, all the constraints, all the limitations you have. And that is the moment when you can sort of somehow guess the flexibility, yes?
Just like a naive question, but can the protein also like move itself?
So you mean that the entire protein sort of walks?
Yeah.
So yes, that does happen.
Flexibility just means that maybe it just wants to move it.
So there are different answers to your question. So one is, yes, it does happen. But it does not happen in the way that but you do see movement like that, just like you see in the ocean. You see that in proteins too. But
It's like because muscles are protein.
yes, and that's a different one. And there you in fact have a different type of movement. But actually part of the muscle movement is actually really a spring, in the sense of a mechanical energy that you store and then you create a spring. Remember I showed you these guns that shoot out proteins into other cells. This is in fact exactly the same mechanism that you use for muscles, where you create a mechanical spring that collapses very rapidly. And that in fact gives muscles the push.
Then we can reload itself, or
yes. The spring is reloading, exactly. And then there is the other typical example that you see for most of the transport in cells. So in the cells, you can imagine they're long tracks. These tracks look like train tracks. when you sort of, if you could make a photo of a cell, you would see fibers or things that look like a spider web through it, or lines of spider webs through it. But the point is that the way proteins move on this is not sort of, I move, but they are pushed forward. It's a train with a cargo, but the tracks are active tracks. So it's a process that involves ATP, and that essentially you have the carriage or the train on it, and then the track pushes the train, and then the next part pushes the train. So the train gets forward and then is pushed further by the track. So it's an active track. So most of the movement really is done by tracks.
Because neurons they also kind of propagate stuff like
neurons.
Like neuron signals.
No, but that is an electric signal.
Okay.
So that is not a mechanical signal, and that in fact does happen. Yes. So proteins carry electrical signals. Essentially, potassium is sort of electrically charged, but they carry direct currents. So they push through electrons, electron transport machines, and are being used for that. Anyway, so one of the first protein structure is a light sensing protein. That in fact is a beta barrel membrane protein. Yes.
Previous slide, was that 44 different instances of the same protein after folding?
Yes. In fact, in this particular case, is supposed to be the same protein measured by NMR, and with the experimental data, 44 different models are compatible with that measurement. So now how do we interpret this this statement? Essentially the interpretation is, in fact, what you show here is 44 possibilities of the way this protein will look.
So can't one do like just one, just let it fold one time and then...
No. But the assumption is that if you let it fold one time today and one time tomorrow, and we go for 44 days, you will see 44 different ones. Yes, we can pick any one of these, or at any time point now, in 10 seconds, again and again and again, 44 of these. So essentially what we assume that this means is we sort of sample the space of possible solutions. So protein structure changes slightly. And that exactly is sort of the dynamic of the protein structure. Where you have movement, that is where you need to be flexible for binding, for instance.
But I guess my question is, after folding, protein will continue to move at certain places that are...
Exactly. That's the idea. So the idea is, so you can read this in different ways. You can read this, I take a protein, again, you cannot measure it this way, but you can think about it as, I take the protein at 44 different time points. Or you can think about, I take the protein now and then I do it again and I see that it still moves. And all of this would sort of essentially give you the same image. Yes?
Which parts are moving kind of depends like how you align it, right? If you look at this one, for example, the aligned parts which are like more dense, are like the ones which are like longer, and like the shorter parts, which are single, they're like, so you would just, and that's probably because like longer ones give a higher alignment score.
You do have an issue here. In fact, now you begin to see why it's so important to get the alignment right. Because if you want to keep this as an interpretation for where the flexible region is, you have to really align that what your alignment makes is something that reflects a majority event. Let's call it this way. Or biophysical event, right? What you do, however, see is there are places where there is no alternative. Right? So no matter what you do with the alignment, this one here is all the same. Right? And then there are places where, in fact, maybe the alignment methods will give you slightly different answers. Yes, true. But anyway, back to the story. We want to predict, and I'm going to cut this one shorter too. Cutting shorter still is too long. In principle, you could predict it, but the problem here, the challenge here becomes that this is a function that does not at all give you a clear cutoff. So if you thought traditional SVMs, for instance, they are binary machines. You want to have something that is flexible and something that is rigid. Where would you put that point here? if you wanted to develop a machine learning device that predicts two states, flexible or rigid, what would be a cutoff? This is these are the raw data here. Where would you put the cutoff? Yeah? So if you do, and I'm sorry for that, thank you very much for the answer. I'm sort of setting you up. Whatever you tell me, I'm going to attack it, okay? I already sort of told you this is difficult, and I also know that nobody answers because I'm going to shoot you down. So thank you for having the courage to be shot down. So you give the answer that most people would give. You put the decision threshold exactly at the peak. There are tons of publications. And this actually has a major problem. Again, most people who develop methods do exactly that. So although I'm going to give an argument why this is really bad, this is what most people do. So you're not at all stupid or anything like that, right? You're in line with many other people. This is so bad because you make mistakes. There are mistakes in the experimental measurement. These mistakes will lead to being, say, some epsilon wrong, right? So some of these things here will be an epsilon wrong. But if you put the decision threshold into the peak, then the ones where the epsilon mistake, no matter how big the epsilon is, makes a difference between one side or the other side is maximum. So whatever epsilon you have, so if the mistake were like this, and now you sort of look at the mistake interval. Unfortunately, I cannot easily do a mistake interval with my, I don't have to, so I don't know how to do that. Let me do it differently. So this is my rendering of the curve that you see. And you see why I use slides instead of self-drawn things. Now this is the epsilon mistake here. And the same interval of epsilon here has a much higher count here than here. Simply by the very definition of the shape. You have a higher count up there, right? If you put it to the maximum, you will put it to the point that is the highest. That's the definition of maximum. As simple as that. And that means that putting a decision onto a maximum makes this decision optimally sensitive to mistakes in the measurement. And that, if you do the next step of the logic, means it's the worst thing you can do. Again, most people do exactly that. But that's a really bad idea, yes?
Yeah, I was just wondering why would anybody do that? Because like the most naive thing would be just look at the area under the curve and tell like make it a 50-50, right? Or if you do like
yes, so now if we had 50-50, that would in fact, so if we had a Gaussian, that would be the same point. Back at you.
Yeah, but at least that like some...
Okay, let's imagine this is a Gaussian. So yes, there is some problem about this particular shape, but if you just imagine a regular Gaussian, still putting it...
That makes sense because...
No, it doesn't. It's the same problem.
Score and you want to guess like who's smarter and stupid.
It makes sense. This is why many people do it. It makes a lot of sense. But
no, obviously, like if like the part of flexible parts is like 10%, it would like do an area under the curve.
it's all true with the area under the curve, what you say. It's all true with machine learning devices like to sort of have even data sets. But it's still the worst thing you can do in terms of, unless you can guarantee that whatever creates your data never makes a mistake. That changes completely the nature of the beast. But actually, there's still a discussion about machine learning then, but let's not enter that because we really don't have time for that. But in a field such as biology, where you really make mistakes in the measurement, most fields you make mistakes. This is a really bad idea. Now, you could argue we put it at a safe point here. But then you have really the problem, and you sort of touched implicitly upon that, you have the problem that there's nothing of the data left. So if you put it, the best in this sense would be to pick one of these extreme points here, but in one of the extreme points, that's why you wanted to have an equal area under the curve, because we have nothing left, yes?
We'll probably split it into three parts, one which is like,
yes,
on the average, one that's extremely rigid, and one that's completely...
So we put one that is a little bit here and one that is a little bit here. That's in fact something that we did. Exactly, but we still predict for that. And this method worked. And I'm going to go through, another way is you predict actually contacts. and you see what is less or well defined, and that allows you to predict disorder too, and then you can sort of predict disorder directly. You can predict disorder only with machine learning. We still don't see it with experiments, and we now know that disorder plays a more important role than we assumed before. In fact, it's so important that we believe it's part of the reason that makes eukaryotes more complex, because there's much more disorder. The fraction of disorder in human is much higher than in the worm, and the one in the worm is much higher than in yeast, and the one in yeast is much higher than in bacterium. And we assume that this has to do with the fact that disorder allows a more complex organization of the cell. 5 more minutes to wrap up 2 other stories. We talked about homology-based inference, we talked about 1D prediction, let's talk about 2D, contacts. So again, 2D is distances between residues. And we could do this as distances, we can do it as contacts. The first question would be, should we zoom into strong interactions, such as contacts between beta strands? And this is in fact one set of methods. Between alpha helices, they're actually not very strong. The ones between the beta strands, they are strong. And that would be a sort of first way of doing it. And there's a bunch of methods available. The way we predict distances or contacts is always long range. So this is the distance map, and I show only the upper diagonal here. Short range essentially is meaning, so close to the diagonal means that you're close to your neighboring residues. And that's why it's short range. You predict binding a contact, a distance in a region that is close to, if you put yourself onto a residue I, that is close to that residue I. Long range means we're looking at things that are further apart. So close would be, we predict whether this one here is contacting that one. Distant would be, I predict whether this one is actually is binding here. That's further apart in sequence. That's what long range and short range means. And it's distinguished with long range and short range because from sequence we want to predict 3D structure. We want the long range ones because we can predict the helices, the secondary structure prediction methods, and now essentially what we want to do is sort of assemble them right. So if we just could have constraints for these helices, maybe then that would be it, right? And again, the contact typically is defined this way. The historical way of doing that is evolutionary coupling, what is today called evolutionary couplings. It used to be called correlated mutations, compensated mutations. All of these words are around. Essentially, the idea is the following. You have an alignment, and here you have protein 1, 2, 3, 4, 5, 6, 7, 8 proteins from, say, 8 different organisms. And highlighted is here one particular column. And you see that in these 3 species there's an R, and these 3 there's a K, and these 3 there's a W. And then you look at the different position in the sequence, and you see also that they sort of have 3 different residues, but what you see is that in fact it's also those 3 that have the same, also those 3 that have the same, and those 2 here that have the same. And if you see something like that, you may suspect that there's a correlation in the event. And what would correlate such an event? Well, essentially, proteins evolve as 3-dimensional structure, as 3-dimensional objects. So if there is a bond between these 2 residues, I and J, bond means they're close in space, then this proximity in space will be maintained in some sense, the way you change. What changes are possible at that position have to do with the fact that they are bonded. So if one, say, would be a positive charge and that is a negative charge, and now one would switch, and the other one would switch, you would maintain the bond, right? But then you would see this as a correlated event in this evolutionary alignment, okay? That's the idea. And that very idea can be used to sort of predict contacts. Here's the first publication of that a while ago, where you see the correlation and the distances. Okay. So what you want is a clear line. It's not at all a clear line. There's no clear signal. Biology means, so computational biologists are often people who say, well, there's a clear signal in here. And there was a long debate over many papers whether there's any signal in here. So there's some signal in here. It took much longer to get it out. This is not completely random. Let's put it like that. And really is not. It's pretty close to random, and it took over 20 years to sort of get the signal out of here. But now, and this is sort of the kind of contact prediction method they got, but now, today, this method in the hands of for instance, Thomas Hopf, I'm going to come back to that later, came to a level where they can actually use that signal and predict protein structures for proteins that had never been observed before. Shown here are 6 membrane proteins. In this particular case, they are predicted and experimental overlay. And in this particular case, all the predictions were done long before the experimental structure was available. So, yeah?
And it's basically just saying, okay, they're like relations, so they have to touch, so they have to be close by.
I'm sorry, what?
Basically what it's saying is like, oh, there's like relations in the residue code, so they might touch.
Yes, and if you predict that particular residues are close in space, then you can use the distance map to really recompute the 3D structure. In that sense, so what you see here as the predicted is really a predicted, all the coordinates are predicted from sequence, essentially from this evolutionary coupling signal. It was originally published in Cell, and it was the first time that people really predicted de novo structures, and these were membrane proteins for complete structures. Here a couple of other papers on that, and the group, just to mention that, so it comes from Debbie Marks and Chris Under. Both of those are now at Harvard. Here's sort of another idea that uses a machine learning device from the olden days and sort of gets some signal. So I cannot remember what is what here, but red is either predicted or observed, and black is the other way around. And you see there's some agreement here. Most likely the red is observed and this one is predicted. So sometimes the prediction method gets it right, and sometimes it gets some signal, and sometimes it essentially gets it wrong. Now, this is just using a simple neural network, and as I already said, there is a breakthrough now through the evolutionary couplings. And that gets us to sort of the next level. What can we do in terms of 3D prediction? One way of doing it is molecular dynamics. Molecular dynamics essentially are energy functions where you sort of compute the energy values and then you fold the protein in the computer. One sort of breakthrough is this project Folding at Home that currently commands 100 petaflops. And sort of classifies it in Wikipedia as world's fastest computing system. Vijay Pandey in Stanford started it. Another sort of amazing example is to model an entire hepatitis B virus, the Schulten group, published last year. And another sort of famous example here is the computer Anton, sort of indicated by this photo here, from D.E. Shaw Research, which is a special purpose computing machine that essentially does only MD and is by far better than any other device that is available. Here is a paper recently published from that group in which they show that prediction method in combination with cryo-EM data and this sort of simulation of MD help to really predict protein structure from scratch. Another example for protein structure 3D can be predicted from sequence came from Google's DeepMind. Google Fold was published was sort of announced in December 2018 at the CASP competition. And the rumor is that they did best at this CASP competition. For every protein for which they worked, there was somebody that was as good as they. So it's not that they are the lonely best. And it's not that they completely nailed the problem, but they are the first that sort of won one of the CASP meetings. So CASP tries to see how good the structure prediction improved over the last 2 years, and that's the first time that an outsider comes and somehow wins one of these competitions. Again, it's not the clearly best method available, and it's not available anyway. It was only available for CASP. So the paper is being written up. We do not know much about the detail. Cast 13 was in December 2018. But clearly it seems there is some sort of breakthrough. The level of performance is not as good as the one that I showed you before, but the proteins from evolutionary couplings that I showed you before unfortunately cannot be applied to all proteins. And this, again, at this point is not available as a prediction method that can be applied to all proteins. So when will we be at the point where this will be available for all human proteins? I do not know. 5 years ago, the moment appeared to be much more in the distance. I believe this is one of the biggest breakthroughs over the last decade. So maybe deep learning pushes us to the moment where we will get very close.